The overall goal of this method is to achieve stable integration of a gene of interest into the human genome of primary cells by the transcriptionally regulated Sleeping Beauty system packaged into integration defective lentiviral vectors.
This method can help answer key questions in gene therapy field, such as integration of the gene of interest into primary cells using Sleeping Beauty system. The major advantages of this technique is that the inducible hyperactive Sleeping Beauty system is efficiently delivering primary cells resistant to several transfection methods. Demonstrating the procedure will be Daniela Benati, a postdoc in my lab.
Prior to the day of transduction, the human primary keratinocytes are grown as described in the text protocol. To detach subconfluent keratinocytes in culture, remove the medium, wash the cells with 1x PBS, and add 1 ml of pre-warmed trypsin working solution to each well. Incubate at 37 degrees Celsius for 15 minutes in the cell culture incubator. Resuspend the cells of a well thoroughly and transfer the cell suspension to a centrifuge tube containing 2 ml of serum-containing medium. Rinse the well once with 3 ml of 1x PBS and add the rinsed solution to the centrifuge tube with the cell suspension. Centrifuge at 580 times G for 5 minutes and discard the supernatant. Wash the cells with 5 ml of 1x PBS.